These are direwolves, predators that died out 12,000 years ago, alive again, born in a lab, running around a private preserve in America. At least, that's what the company that made them told the world. The scientist who looked closer said something very different.
So let's start there and count down the 10 animals we're trying to bring back from the dead.
Counting down from 10 to 1. All right, let's dive in. Number 10, the thylacine, the animal we filmed going extinct. On a freezing night in September 1936, at the Beaumaris Zoo in Hobart, Tasmania, the last known thylacine died. The story goes that a keeper failed to let it into its sheltered den, and it froze. There is film of it, grainy black and white footage of a strange, stiff-tailed animal pacing a concrete enclosure, opening a jaw far wider than any dog's. That footage is part of why this animal haunts people. We did not just drive the thylacine extinct.
We filmed it and then let the last one die, locked out of its own shelter. The thylacine, the so-called Tasmanian tiger, was neither tiger nor wolf. It was a marsupial, a pouched mammal more closely related to a kangaroo than to anything that hunts on four legs elsewhere. And it was the only marsupial apex predator of the modern world, the last member of an entire genus. Through pure convergent evolution, it had grown to look uncannily like a wolf, separated from every real wolf by more than 160 million years of separate ancestry. Tasmanian farmers, blaming it for killing sheep, hunted it out under a government bounty, and the last legal protection arrived only weeks before the final animal died. Since 2022, the de-extinction company Colossal Biosciences has worked with the University of Melbourne's TIGRR lab, led by Professor Andrew Pask, to bring it back. And the science is genuinely advanced.
In 2024, they reconstructed a thylacine genome they say is more than 99.9% complete.
From a 110-year-old head that had been skinned and kept in a jar of ethanol, and recovered long strands of RNA from the specimen's tongue, brain, and eye, the first time RNA had ever been read from an extinct animal. Then they began editing, making more than 300 genetic changes to the cells of the thylacine's closest living relative, a mouse-sized marsupial called the fat-tailed dunnart. They have also reported marsupial reproduction firsts that the whole field lacked, inducing ovulation in the dunnart and culturing fertilized marsupial embryos more than halfway through gestation in an artificial uterus, and are designing an exopouch, an artificial pouch to raise the tiny joey that a marsupial gives birth to after only weeks. Hold on to that size difference, because it frames the honest part. The plan is to edit the cells of a mouse-sized animal toward a wolf-sized predator. Colossal set a 10-year timeline in 2022, which puts a living thylacine past 2030 at the earliest. And even that is optimistic for a marsupial whose pouched reproduction almost no one knows how to engineer. And what emerges, if anything does, will not be the animal in the 1936 film. It will be an edited, dunnart-derived proxy carrying the thylacine's most important traits. The thylacine sits at number 10, not because the science is weak, but because, of everything on this list, it is where the tools to actually attempt this, the genome, the recovered RNA, the most heavily edited animal cells ever made, first genuinely exist. Two details make the loss sting more than almost any other on this list.
The first is the cruelty of the timing. Between 1888 and 1909, the Tasmanian government paid a bounty on more than 2,000 thylacines. And then, in July 1936, it finally granted the species legal protection, about 59 days before the last known one died. We extended our hand to save it almost exactly as the final animal froze in its pen. The second is that the thylacine has never quite been allowed to be dead. In the decades since 1936, there have been thousands of claimed sightings across Tasmania and mainland Australia. A striped shape on a night road, a track in the mud, a photograph that never resolves. None ever confirmed. The animal sits in a strange limbo, extinct in fact and undead in the imagination. And the De-Extinction Project is, in part, an attempt to settle that ache with technology. The animal is not here. The toolkit to chase it is. That is where the climb begins. Number 9. The passenger pigeon.
From the most abundant bird on Earth to zero in a human lifetime. At 1 o'clock in the afternoon on September 1st, 1914, a bird named Martha died on the floor of her cage at the Cincinnati Zoo. She was the last passenger pigeon alive. Her keepers, understanding exactly what her death meant, packed her body in a 300-pound block of ice and shipped it to the Smithsonian. The most abundant bird the planet had ever known was now a single frozen body in a crate. The numbers before the fall are almost impossible to hold in your head. The passenger pigeon may have been the most numerous bird that has ever existed, with a population estimated at 3 to 5 billion, perhaps a quarter to a third of every bird in North America. In 1813, the naturalist John James Audubon watched a single flock pass over Kentucky that took 3 days to go by and blotted out the sun, the droppings falling like snow, the roar of the wings drowning out a man's voice. A single nesting in Wisconsin in 1871 was estimated to cover 850 square miles, and humans took them to zero in about 50 years by industrial slaughter. The railroad and the telegraph meant the location of a nesting flock could be wired to commercial hunters who descended on the colonies, killed the birds by the millions, and shipped them to city markets by the boxcar. At the last great nesting near Petoskey, Michigan in 1878, hunters killed an estimated 50,000 birds a day for nearly 5 months. And the bird's one great strength was also its fatal flaw. It survived predators by sheer overwhelming numbers, breeding only in enormous, dense colonies, which meant that once the flocks were driven below a critical size, the species simply could not reproduce, even where it was protected. There was no slow decline, no last small population. Below a threshold, the whole machine seized.
The effort to reverse this is led by a nonprofit called Revive and Restore, and a researcher named Ben Novak, who has spent more than a decade on this single bird. There is no living cell to clone, and birds cannot be cloned the way mammals can in any case. So the approach is to take the passenger pigeon's closest living relative, the band-tailed pigeon, and edit its genome toward the lost species, then breed the edits into a flock. Even in the best case, the people doing this work estimate that wild, self-sustaining flocks are decades away. Not 2030, but well beyond it. And what they would produce is a band-tailed pigeon engineered to resemble a passenger pigeon, not the bird Martha was. Martha herself was a kind of accidental monument. She had been hatched in captivity and lived her whole life at the Cincinnati Zoo, which at one point offered a $1,000 reward, an enormous sum then, for a single living mate for her.
No one ever claimed it, because there were no more passenger pigeons anywhere to find. In her last years, she shared an aviary with no other member of her species. An old bird trembling on her perch while visitors came specifically to look at the last one. She was probably named after Martha Washington. When she died, a branch of life that had been billions strong within living memory ended on the floor of a cage. And the loss reached past the birds themselves. Into the forest they ran. Billions of passenger pigeons moving in waves were a geological force. They snapped limbs and toppled trees under their collective weight, blanketed the forest floor in droppings, and consumed staggering quantities of acorns, beechnuts, and chestnuts. When they vanished, that entire ecological pressure vanished with them, and ecologists still argue about how the eastern forests rearranged themselves afterward.
A species in the billions is not simply a great deal of one animal. It is a load-bearing wall in an ecosystem, and pulling it out shifts everything that was leaning on it. The shock of watching it happen in real time to the most abundant bird on the continent helped jolt the United States into its first real wildlife protection laws. Too late for the pigeon, but not for everything after it. The lesson is in the arithmetic of the two timelines. It took roughly 50 years to erase 3 billion birds. The attempt to bring back even a faint copy of them has already run longer than that, and the sky is still empty. Number 8, the Aurochs. The wild bull behind every cow, and the men who first tried to rebuild it. In 1627, in the Yaktaro forest of Poland, the last Aurochs died. And we know the date, which is rare, because the animal's end was actually written down. What died there was the wild ancestor of all domestic cattle. A bull standing nearly 2 meters at the shoulder, weighing up to a ton, with long, forward-sweeping horns. An animal painted on the walls of Lascaux 17,000 years ago by people who hunted it. Julius Caesar, describing the wild oxen of the German forests, wrote that they were only a little below elephants in size, of enormous strength and speed, that spared neither man nor beast, and that could not be tamed even when caught young. Germanic warriors mounted their horns in silver and drank from them. Every one of the roughly 1.5 billion cattle alive today descends from this animal, and recent genetics suggests the entire taurine cattle line traces back to a founding group of only around 80 wild aurochs, tamed about 10,500 years ago. We took the most formidable land animal in Europe and bred it down into the dairy cow. The first serious attempt to reverse that is one of the darkest footnotes in the history of biology. In Germany, in the 1920s and 1930s, two brothers, Lutz and Heinz Heck, one running the Berlin Zoo and the other Munich's, set out to recreate the aurochs by breeding modern cattle backward, selecting generation after generation for the size and horns and color and ferocity of the ancestor, trying to reassemble the wild bull from the fragments still scattered in the cattle gene pool. The project became entangled with Nazi ideology. The idea of a resurrected Germanic superbeast roaming a conquered Eastern Empire drew the patronage of Hermann Göring, and some of the resulting Heck cattle were actually released into the Białowieża forest in occupied Poland. They survive in European parks to this day. What the Hecks did, and what their modern successors do, is not genetic resurrection, it is backbreeding, and the distinction is the whole point.
You are not rebuilding the aurochs genome. You are selecting living cattle that happen to resemble their ancestor and concentrating the resemblance. The result looks like an aurochs. It is not one. Modern geneticists have shown the Heck cattle are not even especially close to the real animal's DNA, which is why the more rigorous Tauros program in the Netherlands now works differently, sequencing the actual aurochs genome, identifying which hardy old European breeds carry the most ancestral DNA, and breeding toward the real blueprint as a target, releasing the results into rewilding reserves to take up the aurochs' old job of grazing the wild landscape open. There is a reason the aurochs, of all extinct animals, draws this much effort, and it is not nostalgia for a cave painting. It is ecology.
The aurochs was a keystone grazer, a heavy animal that, in huge numbers, kept the forests of prehistoric Europe broken open into a shifting mosaic of woodland and grassland, grazing back the saplings, trampling paths, churning the soil, creating the patchy, varied landscape that countless other species depended on. When the aurochs disappeared, that wild grazing pressure disappeared with it, and the rewilding movement now sees a robust, self-sufficient, aurochs-like animal as a tool to restore it. A living lawnmower that can be released into a reserve and left to manage the land the way the wild herds once did. That is the real goal of the Tauros program. Not a museum piece, but a working ecological stand-in, hardy enough to survive a European winter outdoors with no human help, and shaped enough like the original to do its old job. Het cattle have already been used this way, with mixed results, in reserves like the Oostvaardersplassen in the Netherlands. But none of that changes what the animal actually is. The aurochs was domesticated not once, but at least twice, in separate events on different continents. The humpless taurine cattle of Europe and the Near East from one wild population, the humped zebu of South Asia from another, which means the genome of the original wild bull is scattered in fragments through different lineages of living cattle all over the world. Backbreeding gathers some of those fragments back together and concentrates the look. It does not reassemble the animal. They are getting closer to the look and the ecological role with every generation. But a resemblance bred out of an animal's own descendants is a portrait, not the sitter. The aurochs that died in that Polish forest in 1627 is gone, and selective breeding, however precise, can only build something that wears its shape. Number 7. The Pyrenean Ibex. The only animal that has ever gone extinct twice.
On January 6, 2000, a tree fell on the last Pyrenean Ibex. Her name was Celia. She was a bucardo, a sturdy, wild mountain goat with heavy ridged horns from the high Pyrenees on the border of Spain and France. And she was, by then, the only one of her kind left. A falling tree in Ordesa National Park crushed her, and with that, the subspecies was extinct. We know the moment precisely because researchers had been tracking her and they found her body. But a year earlier, they had done something with foresight. They had captured Celia, fitted her with a radio collar, and taken a small biopsy of skin from her ear, freezing the living cells in liquid nitrogen. So when she died, the subspecies was gone, but its cells were not. And that set up one of the most extraordinary and least known events in the history of science.
A team led by the Spanish researcher José Folk decided to try to clone the bucardo back, using the same nuclear transfer technology that had produced Dolly the sheep, inserting the nuclei from Celia's frozen cells into goat eggs emptied of their own DNA. The numbers show how far they pushed at the edge of the possible. They built 439 reconstructed embryos. They implanted 57 of them into surrogate goats and goat ibex hybrids. 7 of those surrogates became pregnant, 6 miscarried. And on July 30th, 2003, one surrogate gave birth to a living, breathing Pyrenean ibex, a member of a subspecies that had been completely extinct for 3.5 years. For the first time, an animal that no longer existed drew breath again. It lived for about 10 minutes. The newborn had a catastrophic deformity in its lungs, an extra, solid, useless lobe, and it could not breathe. The scientists tried to save it and could not.
It suffocated in their hands. And so the Pyrenean Ibex earned a distinction that belongs to no other creature in the history of life. It is the only animal to go extinct twice. Once in 2000, when Celia died under the tree, and once again in 2003, 10 minutes after it was reborn. The numbers behind that single birth show how thin the margin really was, and how the bucardo had been failing for a long time before it reached Celia. The Pyrenean Ibex had been hunted relentlessly through the 19th century and squeezed out of its range by livestock, until by the early 1900s, only a few dozen survived in one last stronghold in Ordesa. Protection came, as it so often does, too late to matter. By the 1990s, the count was down to about 10 animals, then fewer, then one. When Fulch's team captured Celia in 1999 and clipped that piece of skin from her ear, they were not so much running an experiment as collecting the last physical trace of a subspecies they already knew they were about to lose. The frozen cells were an insurance policy taken out on a death everyone could see coming. That lung defect that killed the clone was not a freak accident. It is a shadow over cloning itself. Animals made by nuclear transfer suffer abnormally high rates of developmental defects, oversized organs, immune failures, exactly the kind of lung malformation that killed the bucardo. Because reprogramming an adult cell back into an embryo is an error-prone process, and the errors surface in the body that grows from it. Dolly herself was one success out of 277 attempts. The technology that can copy a life can also load the copy with hidden flaws that only appear when the animal tries to live. No one has matched it since, and no project will plausibly produce a living Pyrenean Ibex by 2030. The 2003 clone is history, not a forecast. More than 2 decades later, that 10-minute kid remains the only true de-extinction birth ever achieved. The one time a vanished animal was genuinely brought back to life lies behind the field, not ahead of it, and it lasted the length of a coffee break. But it is the single most important fact in this entire countdown, because it has already answered the question everyone else is still asking. For about 10 minutes on a July day in 2003, extinction was not permanent. The thing we are all taught is final and forever was undone, in a delivery room, by people with frozen cells and a great deal of nerve. And then, 10 minutes later, it was final and forever again. We have already proven extinction can be reversed. We have also already proven how little that may be worth, if what comes back cannot breathe. Number 6. The northern white rhino,
a species kept alive in a freezer. On March 19, 2018, at the Ol Pejeta Conservancy in Kenya, veterinarians euthanized a 45-year-old rhinoceros named Sudan. He had been suffering, and he could no longer stand. Sudan was the last male northern white rhino on Earth. In his final years, he had become a strange kind of celebrity, guarded around the clock by armed rangers, even listed on a dating app as the most eligible bachelor in the world to raise money for his cause. When he died, his subspecies came down to 2 animals, both female, both his direct descendants, his daughter Najin and his granddaughter Fatu. Sudan's life traces the whole tragedy. He was born wild in what is now South Sudan around 1973, captured as a young calf, and spent decades in a zoo in the Czech Republic, where the last captive northern white rhinos were kept. In 2009, in a last gamble, he and 3 others were flown to Kenya and released onto the savannah, in the hope that their ancestral home might trigger the breeding the zoo never produced. It did not work. The other male, Sunni, died in 2014. Sudan died in 2018. A subspecies that had numbered around 2,000 as recently as the 1960s, before poaching for its horn destroyed it, was reduced to a mother and a daughter who cannot give birth. The destruction was almost unbelievably fast and entirely man-made. Northern white rhinos once ranged across parts of Uganda, Chad, Sudan, the Central African Republic, and the Democratic Republic of the Congo. Then, the demand for rhino horn, ground up for traditional medicine in parts of Asia, and carved into status objects, despite being made of nothing but keratin, the same protein as your fingernails, drove an extermination.
Armed poaching gangs cut the population down through the 1970s and 1980s, and the last wild holdouts in Garamba National Park in the Congo were gone by around 2008. A large, ancient animal that had survived for millions of years was effectively shot out of existence in about 3 human decades for a lump of compressed hair. That phrase, "cannot give birth," is the heart of it, and it is precise. This is what scientists call functional extinction. The species is not technically gone. There are 2 living animals grazing under guard in Kenya. But Najin has dangerous problems in her hind legs, and Fatou has a uterine condition that makes pregnancy impossible. There is no male. The northern white rhino, in every functional sense, is already over, even though 2 of them are still alive. Except for what is in the freezer. An international consortium called BioRescue, led by the German scientist Thomas Hildebrandt, has spent years on a methodical rescue.
Sperm from the last males was collected and frozen before they died. From Fatou, the team has harvested egg cells more than 20 times in delicate operations under anesthesia, flying them to a laboratory in Italy run by the reproduction specialist Cesare Galli, where they are fertilized by injecting the frozen sperm directly into them. By early 2026, they had built 39 pure northern white rhino embryos, frozen in liquid nitrogen at -196 degrees. An entire species' future suspended in steel tanks. The plan is to implant them into surrogate mothers of the closely related southern white rhino. They proved that surrogacy works when a southern white rhino became pregnant from a transferred embryo in 2023. Though that surrogate, named Kura, died of an infection before the calf was born.
But the transfers of actual northern white rhino embryos, 6 of them, by the spring of 2026,
have still not produced a single lasting pregnancy, and the team has had to introduce new surrogate females and keep trying. As of this writing, there is no calf, and no guarantee there will be one. And in parallel, scientists in Japan are trying to turn ordinary preserved skin cells into rhino eggs and sperm, which matters enormously because a species rebuilt from 2 females and a few dead males would be perilously short of the genetic diversity it needs to survive. This is what makes the northern white rhino different from everything else on this list, and the most realistic candidate to produce a living calf by 2030. This is not a fossil or a century-old head in a jar. The species is alive right now. In 2 animals you could go and watch eat grass, and in 39 embryos in a tank. No vanished genome has to be reconstructed from scraps. The entire race is just to move those embryos the last few centimeters, from the freezer into a working womb, before the window closes and the last 2 living individuals are gone. The future of a species is sitting complete in a tank of liquid nitrogen. The hardest thing in the world is getting it out. There is one reason to believe this can actually work, and it is grazing a few enclosures away. The southern white rhino, the northern's close cousin, was itself reduced to fewer than 100 animals in the late 1800s, hunted to the brink, and through a century of dogged conservation, it recovered to around 20,000, the single greatest comeback in rhino history. That recovery is why there are southern white females available to serve as surrogates at all. The rescue of the northern is, in effect, an attempt to compress that century of work into a laboratory, starting from a far worse position. But the worst position is real. A species rebuilt from the eggs of one female and the frozen sperm of a few dead males would be desperately inbred, which is exactly why the parallel work in Japan, turning ordinary preserved skin cells into fresh rhino eggs and sperm, matters so much. Without new genetic diversity conjured out of those banked cells, even a successful calf would be the start of a population too narrow to last. Getting one rhino born is the visible race. Getting a viable future for the species is the quieter, harder one underneath it. Number 5. The moa, the tallest bird that ever lived, funded by Hollywood. Whew.
The South Island giant moa stood up to 3.5 meters tall, around 12 feet, when it stretched its neck to full height and weighed as much as 230 kilograms, more than 500 pounds.
It was the tallest bird that has ever existed. A flightless giant that strode through the forests of New Zealand with no wings at all. Not stunted wings, not hidden wings, but no wing bones whatsoever. The only known bird to have lost the four-limb skeleton completely. There were 9 species of moa, from turkey-sized up to this towering thing. And for millions of years, they were the dominant plant eaters of New Zealand, a land that until very recently had almost no land mammals. The moa filled the role deer and cattle fill elsewhere. A quick correction, because the internet loves to claim the moa could disembowel a person. It could not and did not. The moa was a herbivore that ate leaves, twigs, and fruit. The reputation for slashing attackers belongs to a different bird, the cassowary. The moa's danger ran in the other direction, toward it. Its biology was strange in its own right. The females were so much larger than the males, up to 1.5 times the height and nearly 3 times the weight, that 19th century scientists sorted the two sexes into entirely separate species, an error that stood for over a century until DNA testing corrected it. And the moa was not even at the top of its world. Above it flew Haast's eagle, the largest eagle that has ever existed, with a wingspan near 3 meters and talons like a tiger's claws, which hunted the giant moa by striking it at speed and crushing its pelvis. Then, humans arrived. When the Polynesian ancestors of the Maori reached New Zealand around 700 years ago, they found a land of huge, slow, flightless birds with no fear of a ground-walking hunter.
The archaeology, the butchery sites, the earth ovens full of moa bone, the vast middens of eggshell, combined with population modeling, indicates that all 9 species of moa were hunted to extinction within roughly 150 years. One of the fastest megafauna collapses on record, carried out by a relatively small human population. And Haast's eagle died with them, having lost the only prey large enough to sustain it. In July 2025, the moa became one of the highest-profile de-extinction projects in the world, and the reason was the name attached to it. The filmmaker Peter Jackson, director of the Lord of the Rings films, a New Zealander who has privately collected 300 to 400 moa bones over the years, is bankrolling the effort along with his partner, putting in millions alongside Colossal and, crucially, the Ngāi Tahu Research Center, representing the Maori of the South Island, who are directing the work.
That indigenous leadership is unusual and deliberate, framing the project as the return of an ancestor as much as a feat of engineering. For Ngāi Tahu, the moa is not a curiosity. It is woven into the history of how their ancestors lived and what they lost. And putting Maori knowledge and authority at the center of the work is partly an attempt to do de-extinction differently from the start, as a cultural act, not just a corporate one. But that framing also raises the question the whole project has not answered, which is what a moa would even come back to. New Zealand today is not the New Zealand the moa ruled. The forests it browsed have been cut and replaced. The predators that would eat a young moa now include stoats, cats, and dogs that never existed there before. Haast's eagle, the only thing that ever hunted an adult, is long gone. A bird bred to be released has to have somewhere to be released into, and the moa's world was dismantled centuries ago.
You could, in principle, build a large flightless bird and keep it in a fenced sanctuary.
Whether you could ever restore the moa to a wild it no longer has a place in is a different and much harder thing. But the honest flag is about where this actually stands, which is still early, though no longer quite as early as a hard drive. The project is working through the genomes of all 9 moa species, aiming to finish the first
the South Island giant, in the summer of 2026, comparing them to living birds to find which genes matter. And in May 2026, the team announced a genuinely clever piece of the puzzle: an artificial egg, a transparent, 3D printed lattice that behaves like an eggshell, letting oxygen pass evenly across the membrane so an embryo can develop in full view inside an incubator, solving a problem that wrecked earlier artificial egg designs. As a proof of concept, they hatched 26 ordinary chicks from it.
That matters because a moa egg is enormous, about 240 millimeters long, roughly 4 times the length of the egg of its nearest living relative, and there is no living bird remotely large enough to lay or incubate one, so any moa-like creature would have to be hatched by machine. Because here is the deeper problem the genome work keeps running into. The moa's closest living relative is not another giant. It is the tinamou, a partridge-sized, ground-dwelling South American bird. A bird the giant moa outweighed perhaps 9-fold, that last shared an ancestor with the moa tens of millions of years ago, far further back than the gray wolf is from the dire wolf. There is a strange irony folded into that. The tinamou can fly, which means the moa's nearest surviving cousin never lost the wings the moa threw away entirely.
To rebuild a wingless 12-foot giant, you would be starting from a small bird that still takes to the air and hatching the result inside a printed plastic egg. Colossal's chief executive says a moa could walk again within a decade. New Zealand's own leading ancient DNA experts have been blunt that you cannot truly de-extinct the moa, that the most you could build is a large, flightless, gene-edited bird that resembles one. So this is the shape of the most glamorous project on the list: a printed egg that can hatch a chicken, a tinamou genome, the first moa genome still months from finished, and a Hollywood director's promise: real accelerating machinery aimed at a giant that for now exists only as bone, ancient DNA, and ambition. Number 4. The Gastric-Brooding Frog, the animal that gave birth through its mouth.
In 1973, in the rainforest creeks of southern Queensland, Australia, biologists described a small, brownish, easily overlooked frog called Rheobatrachus silus. Then, someone watched a female reproduce, and the entire field of biology had to sit down. What this frog did is so strange that in fiction you would dismiss it. The female gastric-brooding frog swallowed her own fertilized eggs. She took them into her stomach, the organ whose entire purpose is to dissolve food in acid, and then she shut the stomach down. The developing eggs released a hormone that switched off her production of hydrochloric acid and stopped the organ from churning, converting her stomach into a womb. She stopped eating for around 6 weeks while her young grew inside it, until they filled her body cavity and pressed on her lungs so hard she had to breathe partly through her skin. And when they were ready, she gave birth the only way she could. She opened her mouth and brought them back up, burping out fully formed live froglets. A mother vomiting her own living children into the world. No other animal known to science has ever gestated its young in its stomach. The discovery was almost an accident of timing. The southern species was first described in 1973 by the biologist David Liam, and it fell to the Australian frog specialist Michael Tyler and his colleagues to work out what it was actually doing. Watching in disbelief as a female that had swallowed her eggs later opened her mouth and produced fully formed froglets. The mechanism turned out to hinge on a single chemical signal, a prostaglandin released first by the eggs and then by the developing young, that ordered the mother's stomach to stop making acid and stop emptying itself for weeks on end. Then, in 1984, just as researchers were beginning to study the first species, a second one was discovered a few hundred kilometers north, the northern gastric-brooding frog, which did the same impossible thing by a slightly different route. For a brief window, science had two of these creatures to learn from. Within a couple of years, it had none. Both were gone almost before anyone had grasped what was being lost. We lost it almost as soon as we found it. The southern gastric-brooding frog was declared extinct by the mid-1980s, barely a decade after its discovery, and a second species, found in 1984, vanished within a year or two of that. The likeliest cause is chytrid, Batrachochytrium dendrobatidis, a fungal disease that attacks the skin amphibians breathe and drink through, and which over the past half-century has become the single most destructive wildlife disease ever recorded, driving the decline of more than 500 amphibian species and the outright extinction of around 90. The gastric-brooding frog, in its cool mountain streams, appears to have been one of its earliest victims, gone before science understood what was killing the world's frogs.
There is a particular bitterness to this one, beyond the animal itself, and it is medical.
The way the frog shut off its own stomach acid, safely, on a biological command, and then switched it back on, was of intense interest to researchers studying human ulcers and acid disorders. Whatever that frog knew, chemically, about controlling the most destructive process in the human stomach, it took with it.
In 2013, a team at the University of New South Wales, led by the paleontologist Mike Archer, tried to bring it back in a project they named Lazarus. Using frozen tissue a researcher had stored decades earlier, they inserted the dead frog's cell nuclei, by the same Dolly method, into the eggs of a living relative, the great barred frog, that had been emptied of their own DNA. And something happened. The eggs began to divide. They reached an early embryonic stage, a tiny ball of cells, and DNA tests confirmed those cells carried the genome of the extinct gastric-brooding frog, not the host. For a few days in a dish in Sydney, the animal existed again as living tissue. Then every embryo stopped, none reaching even the tadpole stage, halted by some defect in the reprogramming that no one has overcome. No living gastric-brooding frog has ever been produced.
The frog is worth dwelling on because it is not an isolated tragedy. It is the leading edge of the largest extinction crisis on the planet that almost no one talks about. Amphibians are vanishing faster than any other group of vertebrates, and chytrid is the main reason. A fungus probably spread around the world by the global trade in frogs, which has now been detected on every continent they live on, and has driven the worst disease-caused loss of biodiversity ever recorded. Whole communities of frogs in the mountains of Central America and Australia collapsed within a few years of its arrival, often before anyone had even named the species being lost. The gastric-brooding frog was simply one of the first to go, in a wave that is still moving.
Which is why, alongside the headline de-extinction projects, there is a quieter and arguably more important effort underway. Biobanking. Frozen arcs of cells, tissue, eggs, and sperm from endangered and recently extinct animals, stored in liquid nitrogen against a future when we might know how to use them. The whole Bucardo experiment and the rhino's frozen embryos only exist because someone had the foresight to freeze cells before it was too late. For thousands of species now sliding toward the edge, that frozen tissue may be the only piece of them that survives long enough for the technology to catch up. The frog leaves a hard measure of the distance we still have to cross. Here was an animal that turned its stomach into a womb as a matter of routine, and we could not keep a cluster of its resurrected cells alive for a single week. The gap between editing a genome in a slide and building a living, breathing, functioning animal is enormous, and this is where it becomes a wall.
Number 3. The dodo, the icon of extinction itself. No animal is more synonymous with extinction than the dodo. Dead as a dodo is how the language itself files the idea of something gone forever, and the bird has become a cartoon, Fat, stupid, comic, practically begging to die out. Almost none of that is true, and the gap between the real dodo and the joke is the first thing to fix. The dodo was a large, flightless pigeon that lived on the island of Mauritius in the Indian Ocean and nowhere else. It stood about a meter tall and weighed somewhere around 15 to 20 kilograms, roughly the size of a large turkey. It was flightless and ground nesting because, for the whole of its history, Mauritius had no predators that could threaten it, so it had no need to flee and no instinct of fear. It was not stupid, it was fearless, which is a very different thing, and which turned fatal the moment the world changed. When Dutch sailors arrived around 1600 on what had been an uninhabited island, they brought rats, pigs, monkeys, and dogs that ate the dodo's eggs and chicks straight off the ground. And within roughly 80 years, the bird was gone, last reliably seen in the 1660s. The sailors did eat dodos, though they often found the meat tough. The real killers were the animals that came off the ships with them. And the extinction happened so casually, so beneath anyone's notice, that no European thought to preserve a single complete specimen. By the time naturalists understood the bird was gone forever, there was almost nothing left of it to study, which is part of why it took until the genetic age to even settle what kind of bird it had been. It did not die of stupidity. It died of having evolved in a world without us, and then meeting us. The fat, waddling caricature comes largely from paintings of overfed, captive birds shipped to Europe. Analysis of dodo skeletons suggests an animal that was actually reasonably athletic, well adapted to its forest. And here is the fact that resets the whole bird. The dodo's closest living relative is the Nicobar pigeon, a small, iridescent bird you could hold in two hands. The dodo was a giant pigeon, and its nearest relative of all was the Rodriguez solitaire, another large flightless pigeon from a neighboring island that humans also drove extinct. The icon of extinction was not a lonely freak. It was one of at least two giant island pigeons we erased. The dodo is now a flagship project of colossal biosciences, with the paleogeneticist Beth Shapiro, who in 2022 sequenced the dodo's full genome from a specimen preserved in Copenhagen. But the dodo is the hardest case in the company's entire portfolio, and the reason is bird biology.
You cannot clone a bird the way you clone a mammal. There is no egg cell you can simply slip an edited nucleus into, because a bird embryo develops on the surface of an enormous yolk inside a hardening shell. Instead, you have to edit primordial germ cells, the cells deep in an embryo destined to become eggs and sperm, grow them, and channel them through living surrogate birds across 2 generations. By 2025, Colossal had made real progress on exactly this. They grew pigeon primordial germ cells for the first time and engineered sterile chickens ready to carry edited cells, which the team called a pivotal step. They have also established a breeding colony of Nicobar pigeons in Texas to supply the cells, and formed a Mauritius Dodo Advisory Committee to plan any eventual rewilding. But they remain, in their own words, years from a living bird, and bird de-extinction is by a wide margin the least likely on this list to walk by 2030.
A few things are worth knowing about what we are actually working from, because the dodo's remains are heartbreakingly thin. There is no complete stuffed dodo anywhere on Earth. The single most important relic is the Oxford dodo, a mummified head and a foot at the Oxford University Museum of Natural History, the only soft tissue of the species left in existence, and the source of much of what we know about it. From scraps like that, and the Copenhagen specimen, the whole genome had to be reassembled. And the bird is so entangled with myth that even its ecology became a fable. For years, a popular story held that a Mauritian tree, the Tambula coke, could no longer reproduce because its seeds only germinated after passing through a dodo's gut, so that the tree was supposedly dying out alongside its lost disperser. It is a lovely story, and almost certainly wrong. The trees germinate fine without dodos, but it shows how readily we drape this animal in meaning it never carried. The money and attention now pointed at it show how potent that meaning still is.
Colossal has raised hundreds of millions of dollars and been valued in the billions.
Its backers reportedly even include the venture arm of the CIA, drawn to the underlying genetic tools. The company frames the Dodo project partly as conservation, promising to work with Mauritius to restore the island's degraded ecosystems. And there is a real argument that the techniques developed chasing the dodo could help save living birds. But that promise runs into the same problem the moa faces. The dodo's world is largely gone. The Mauritius the dodo evolved on, predator-free, densely forested, utterly safe, no longer exists. The island has been heavily deforested and overrun with exactly the introduced rats, monkeys, and other animals that drove the dodo extinct in the first place. The same threats that menace Mauritius's surviving native birds today.
A resurrected dodo could not simply be set loose into the forests that made it. Those forests and that safety were destroyed centuries ago. It would need a protected, managed refuge built and defended for it, which raises the obvious question of why not spend that effort defending the living species clinging on there right now. But it is hard to ignore that the dodo is also the perfect logo. The universal symbol of extinction, the saddest bird in the world, the one whose return would generate the most headlines. The science is being pulled, as it is everywhere on this list, toward the animal that markets best. There is a dark symmetry in that. The single most famous symbol of extinction's permanence, the bird whose name is shorthand for gone forever, turns out to be one of the hardest things on Earth to bring back. And its nearest living echo is a pigeon, the most ordinary bird imaginable, the kind most people would step over on a sidewalk without a glance. Number 2.
The direwolf. The moment a company said it had done it, on April 7th, 2025, Colossal Biosciences announced to the world that it had brought back the direwolf, a predator extinct for more than 12,000 years, and that 3 of them were alive at that moment on a 2,000-acre preserve in the United States. Their names were Romulus and Remus, 2 males born on October 1st, 2024, and Khaleesi, a female born that January. The direwolf was a real Ice Age apex predator of the Americas, and thanks to Game of Thrones, where the Stark children each raised one, it was also a pop culture icon. The story wrote itself. The first animal ever resurrected from extinction, and it was a creature from the most famous fantasy series on television. Photographs of 3 snow-white wolves flooded every feed on Earth. This is the biggest honesty moment in the countdown, so here is exactly what happened, because it is genuinely impressive and also not what the headline said. Colossal sequenced the dire wolf genome from ancient DNA, a 13,000-year-old tooth, and a 72,000-year-old skull. They compared it to the gray wolf, identified the genetic differences they judged most important for the dire wolf's size, build, and pale coat, and made 20 edits across 14 genes in gray wolf cells. No ancient dire wolf DNA was spliced in. The genes were rewritten to match. They grew embryos from those edited cells, implanted them in surrogate domestic dogs, and of 4 pups born, 3 survived. By their first birthday, Romulus and Remus weighed over 120 pounds each. By early 2026, all 3 were full-grown and, for the first time, living together as a single pack on the preserve, eating, playing, and attempting to hunt together, which the company presents as evidence the animals behave like the predator they were modeled on. Now the part the headlines skipped.
The overwhelming consensus among independent scientists is that these are not dire wolves. They are gray wolves with 20 edits. A 2021 study in Nature had already shown that the direwolf was not even closely related to the gray wolf. The two lineages split around 5.7 million years ago, far enough apart that the direwolf was reclassified into its own genus, Enocian, and almost certainly could not have interbred with wolves at all. They looked alike through convergent evolution, not kinship. We know the real animal intimately too. From the La Brea tar pits in Los Angeles alone, paleontologists have recovered more than 4,000 direwolf individuals, animals that died lunging after prey already trapped in the asphalt, and that vanished around 13,000 years ago when the giant Ice Age mammals they hunted disappeared.
You cannot bridge nearly 6 million years of separate evolution across countless genes with 20 edits.
What Colossal made is a gray wolf engineered to resemble a dire wolf, a striking feat, and not a resurrected species. The real dire wolf is worth picturing because it makes the gap concrete. It was not simply a big gray wolf. It was a differently built predator, heavier through the chest and shoulders, with a more massive skull and jaws engineered to crack the bones of the megafauna it brought down. Horses, bison, ground sloths, the young of mastodons. La Brea preserves them in such numbers because packs waded into the tar to feed on already trapped prey and were caught in turn, layer upon layer, until the direwolf became the most common large mammal in the entire deposit. These were animals shaped, down to the bone, by an ecosystem of giants that no longer exists. The white wolves on Colossal's preserve will never crack a mammoth's femur, because there are no mammoths, and because their jaws are gray wolf jaws. To get the real animal's body, you would have to rebuild not 20 genes, but the thousands that diverged over those millions of years. And even then, you would have the body without the world that gave it a purpose. This is where the company and its critics are really fighting, and the disagreement now has a name. Colossal calls what it does functional de-extinction, recreating the appearance and ecological role of a lost animal, rather than an atom-for-atom copy. And by that definition, an edited gray wolf that looks and behaves dire wolf enough might qualify. Most independent biologists reject the framing, because the public does not hear the word functional. The public hears the dire wolf is back, which is simply not true. The fight is not pedantic. It decides what the word de-extinction is allowed to promise, and whether a company is permitted to sell 20 edits to the world as a resurrection.
Underneath the marketing fight is a genuinely deep question with no settled answer. What is a species? And how much of one do you have to rebuild before you can say you have it back? Change 20 genes in a gray wolf, and it is obviously still a gray wolf. Change 20,000 until the whole genome matches, and almost everyone would agree you had a dire wolf. Somewhere in between, the answer flips, and no one can say where. Because a species is not only its DNA, it is a population with a history, a way of life, behaviors passed from parent to pup, a place in an ecosystem that no longer exists. The direwolves of the Ice Age hunted prey that is also extinct, in a climate that is gone, in packs whose knowledge died with them. Even a perfect genetic copy would be an orphan in the wrong world. And the stakes are not only philosophical.
The announcement itself was a masterclass in spectacle. The company put the white pups on the cover of Time magazine with the word extinct crossed out, leaned hard on the Game of Thrones connection. The series author, George R.R. Martin, is an investor and was thanked by name, and named the animals after the founders of Rome and a Targaryen queen. It was engineered to be irresistible to the media, and it worked. Underneath the show, though, the same pipeline was doing something quieter and genuinely valuable. Colossal also cloned red wolves, a critically endangered species native to the American Southeast, with only a few dozen left in the wild. Using cells from a line of wild ancestry, that could add badly needed genetic diversity to the survivors. That is real conservation, and it tends to get a fraction of the attention the imaginary direwolf received.
But figures in the United States government also cited the direwolf announcement while discussing the weakening of endangered species protections, on the logic that if extinction can simply be reversed, it matters less. That is precisely the danger the field's own critics warn about. The first animal sold to the public as brought back from the dead is, underneath the fur, a question nobody has answered, and a tool that can be used to argue we should care less about the animals still slipping away. Number 1. The woolly mammoth. The Holy Grail and the mouse that came first. On March 4, 2025, Colossal Biosciences introduced the world to a mouse. It was small and golden brown and astonishingly fluffy, with hair about 2 centimeters long and curly whiskers, and it was one of the most consequential lab animals ever made. They called it the woolly mouse, an ordinary lab mouse, gene-edited with a suite of traits drawn from the woolly mammoth's evolutionary playbook: a thick woolly coat, an altered fat metabolism, and cold tolerance genes, all engineered in at once. The mouse was not the goal. It was a dress rehearsal, a proof that the company could stack multiple complex mammoth-like traits into a single living, healthy animal. The goal is the most ambitious de-extinction target on Earth: the mammoth itself. The mammoth is the Holy Grail, and the animal that began this entire modern movement. Woolly mammoths roamed the cold steppe of the last ice age, 11 feet at the shoulder, clad in dense fur, with small ears and a hump of fat against the Arctic. The last of them died only about 4,000 years ago on Wrangel Island off the Siberian coast, which means there were living mammoths on Earth when the pyramids of Egypt already stood.
And because they died in the cold, their bodies were preserved in permafrost, some so intact that researchers have recovered flesh, hair, and readable DNA from animals tens of thousands of years dead. That preservation is why the mammoth, of all the lost giants, is the one people believe they can actually rebuild. Siberia keeps giving them up. Baby mammoths like the calves known as Luba and Yuka, frozen for tens of thousands of years, with their skin, their fur, even their last meal still in their stomachs, intact enough to look asleep. From bodies like those, researchers have read the mammoth genome in extraordinary detail, which is exactly what makes the gap between knowing the genome and building the animal so visible. There is a poignant footnote written into that genome too. The very last mammoths, the ones that clung on for those final millennia on Wrangel Island after the mainland herds were gone, were a tiny, isolated population. And their DNA shows the damage of it: an accumulation of harmful mutations and a collapse in genetic diversity. The molecular signature of a species inbreeding its way toward the end, even before the last individual died. The mammoth did not simply vanish. On that last island, it slowly came apart from the inside, which is a sobering thing to hold next to a plan to rebuild it from the genome of one closely related elephant. The dream behind it is bigger than nostalgia, and it is the engine of the whole company. The modern effort traces to the Harvard geneticist George Church, who began working on mammoth genes around 2015 and co-founded Colossal in 2021. The stated goal is ecological. The idea, championed by the Russian scientist Sergei Zimov and his Pleistocene Park in Siberia, that large grazing herbivores once kept the Arctic as grassland, the so-called mammoth steppe, and that restoring such animals could trample snow, knock down trees, and help keep the permafrost frozen, slowing the release of the immense stores of carbon locked beneath it. Whether engineered elephants could ever do that at scale is deeply uncertain, but it is the argument that turns a de-extinction spectacle into something its backers can call climate science. The stakes it gestures at are genuinely vast. The Arctic permafrost holds on the order of twice as much carbon as is currently in the entire atmosphere, locked in frozen ground that is now beginning to thaw. and anything that slowed that thaw would matter enormously.
The objection is that the path from one edited elephant to stabilized permafrost runs through a chain of enormous, unproven assumptions. That the animals would thrive in the Arctic, breed into vast herds, and reshape the tundra the way mammoths supposedly did. None of which has been shown, and all of which would take many decades and many thousands of animals even to begin to test. The plan is precise and enormous. The mammoth's closest living relative is the Asian elephant, which shares around 99.6% of its DNA, the two having diverged about 6 million years ago. In 2024, Colossal created induced pluripotent stem cells from the Asian elephant, reprogrammed cells that can be edited and grown, the raw material for the whole pipeline. The team is working through a list of roughly 85 genes it believes encode the mammoth's key traits, editing them into elephant cells, with the aim of building an edited embryo, implanting it into an Asian elephant surrogate, a pregnancy of about 22 months, and producing the first mammoth-like calf by the end of 2028. The woolly mouse already proved the gene stacking works in a living mammal, editing several genes at once, including ones that govern hair length, texture, and color, and others that change how the body handles fat, to produce that long, golden, wavy coat. That mouse colony now numbers in the hundreds, with the edits passing intact to each new generation. To build toward the real thing, the company has assembled more than 50 woolly mammoth genomes spanning over 1 million years of the animal's history. And as of early 2026, it still publicly targets a first calf by 2028. And in late 2025, to strengthen the pipeline, Colossal bought the world's leading animal cloning company. But the honest frame on the Holy Grail is the same one that runs through this entire list. The mammoth is the closest thing here to a real by 2030 candidate. There is a concrete pipeline, a concrete date, and genuine breakthroughs behind it. But what would be born, if the date holds, is not a woolly mammoth. It is an Asian elephant, gene-edited to be cold-adapted and hairy, carrying dozens of mammoth traits in an elephant genome. It would be magnificent, and it would not be the animal that died on Wrangel Island. There are deep problems still unsolved, and they are not just technical.
The surrogate would be an Asian elephant, an endangered species in its own right, with a slow, difficult, poorly understood reproduction, used as the experimental womb for a vanished animal across a near 2-year pregnancy, with all the risk that carries for a creature we are simultaneously trying to save. The artificial womb floated, as the humane alternative does not exist for any large mammal, and may not for a very long time. And even past all of that lies the question almost no one in the headlines asks. Then what? One calf is a press release. A species is a herd, breeding across generations, with somewhere to live. To make a real population of mammoths, you would need many animals, many surrogate elephants, decades of work, and a wild place cold enough and empty enough to put them. And at the end of it, you would still have cold-adapted elephants, not the thing that died.
Prominent mammoth scientists, including specialists who have spent careers reading mammoth DNA from the permafrost, call the 2028 date marketing more than biology, and point out that even a successful calf would be a single cold-adapted elephant with no herd, no mammoth mother to learn from, and none of the social knowledge a real mammoth needed to survive. The paleobiologist Tori Harridge, who studies mammoths, has been among the public skeptics, noting that an elephant edited to be hairy is not the same as restoring a species, and that the ecological promises run far ahead of the evidence. Mammoths were intensely social animals that learned migration routes, feeding grounds, and survival itself from older females across decades. A lone engineered calf raised by elephant keepers would have none of that. And one calf, however hairy, is not a herd, a culture, or a functioning population on the steppe.
The timeline has already slipped once. The company originally floated a thylacine by 2025, a date that came and went, which is the whole story of de-extinction, compressed into one animal. We may, by the end of this decade, put a woolly, cold-adapted, gene-edited elephant on the Arctic steppe. We will call it a mammoth, and the argument over whether it really is one will almost certainly outlast the calf. Whew. Finale. 10 animals, 10 attempts to do the one thing nature has never permitted, to make extinction stop being permanent. Now line them up, because side by side they tell a story no single headline does. Not one of these projects has resurrected the original species. Whew. Not one. The Pyrenean Ibex was genuinely cloned back and died in 10 minutes, the only creature ever to go extinct twice. The dire wolves are gray wolves with 20 edits. The mammoth will be an edited elephant. The thylacine will be an edited Dunnart.
The moa, the dodo, the passenger pigeon, the gastric-brooding frog are still genomes on hard drives, or clusters of cells that stopped dividing, with no living animal at all. The Aurochs is a look-alike bred out of cattle, and the northern white rhino, the one closest to a real birth, is not a reconstruction at all. It is a still living species being pulled back from the edge by IVF. Across the whole list, the phrase "brought back from extinction" is doing far more work than the science beneath it can yet support. Two honest problems sit under everything. The first is that a genome is not an animal. Even a perfect edit gives you a body, not a being. Not the behavior a mammoth calf would have learned from its herd. Not the gut microbes the species carried. Not the migration routes, or the calls, or the culture. You can build the hardware. The software died with the last individual.
What comes back is a living creature shaped like the lost one, released into a world that no longer has a place built for it. The second problem is sharper. Money and attention are finite, and every animal on this list was chosen by someone over the thousands of others vanishing right now. The mammoth, the direwolf, the dodo, the moa. These are the celebrities of extinction, the ones that raise hundreds of millions of dollars and make magazine covers. Meanwhile, the species actually winking out today, in the largest extinction event since the asteroid, are mostly small and obscure. Frogs, freshwater mussels, insects, plants, the unglamorous machinery that holds ecosystems together. There is a real fear among scientists that de-extinction becomes a permission slip, that if the public believes extinction is reversible, the urgency to prevent it quietly drains away. Remember that the surrogate mother proposed for the woolly mammoth is the Asian elephant, an endangered animal in its own right. We are talking about using a vanishing species as the womb for a vanished one. And yet the thylacine sat at the hopeful end of this list for a reason. The tools coming out of all this are real, and they are already turning back toward the living. The assisted reproduction techniques built for the northern white rhino are being adapted for other endangered species. The marsupial reproductive science from the Thylacine project is being aimed at saving the Tasmanian devil from a contagious cancer. The most honest case for this entire field may not be the mammoth or the dire wolf at all. It may be that the byproduct, better genetics, better stem cells, better ways to keep a dwindling species breeding, ends up saving more living animals than the headline resurrections ever bring back dead ones. It is worth being honest, too, about what we are really chasing when we try to resurrect the dead. Because it is not only science. There is grief in it. The wish to undo a specific loss, to reach back and fix the thing our species so plainly broke.
There is guilt, the sense that having caused these extinctions, we owe the animals a return ticket.
And there is something less noble, the thrill of godlike power, of being the generation that finally beat death at the level of the entire species. None of those impulses are illegitimate, but they are not the same as a clear-eyed plan for a living animal in a real ecosystem. And when a company can raise hundreds of millions of dollars on grief and spectacle, the temptation is always to announce the resurrection before the biology can honestly support it. To sell the feeling of bringing something back, which is easy, in place of the staggeringly hard work of actually doing it. The headline is the cheap part.
The animal is the expensive one. So picture where de-extinction actually stands, right now, today. Not a mammoth thundering across the tundra. In a conservancy in Kenya, 2 northern white rhinos, a mother and a daughter, graze under 24-hour armed guard. They are the last 2 of their kind, and neither can give birth. Their species' entire future does not live in them. It lives a continent away, in a steel tank in an Italian laboratory, as 39 frozen embryos suspended in liquid nitrogen.
The one-way door that has stood for 4 billion years finally has people pushing on it.
We still do not know if it opens. If this kept you to the end, subscribe. This story is moving fast, and we will keep following what is real and what is hype as the calves, or the headlines, actually arrive.
And in the comments, tell me 2 things. Which of these 10 did you not know we were trying to bring back? And the harder one, if you could resurrect exactly one, the mammoth, the thylacine, the dodo, the moa, the passenger pigeon that once darkened the sky, which would you choose? And is bringing it back the right thing to do at all? Until next time.